Thanks so much for this opportunity. So I'm from the Africa Health Research Institute, so AHRI, in our Leslie's lab. And yeah, we've been basically, we've been lucky enough to have this CytAssist instrument, and it's the first in Africa. And what we are basically looking at, sorry, it's not transitioning. Here we go. So we've established the CytAssist instrument and this workflow in our lab, and that's also with thanks to our funders and also to our collaborators at Colorado State University. And what this has allowed us to do is implement Visium HD in our lab, and this allows you to look at spatial transcriptomics at a single cell resolution. So this is just one of the examples of data that we have generated. So what I want to do is just walk through our workflow, and then I'll accept any questions afterwards. So we're wanting to assess, so we're focusing on TB, and we're wanting to look at the host-pathogen interaction at the sites of disease in the human lung. What I do also want to include is a second approach that we've taken in using tissue microarrays, and this is from a different study using gut biopsies. But this is just also a nice approach in which you can actually multiplex the output from the Visium HD capture slide. So that is just an example that I want to show, but I'll focus on the TB results that we have so far. So I'm going to focus on this particular PID. So this is the H&E for the sample that I'll focus on, and then get into the workflow. So this is essentially how the method works, which I'm sure everyone is aware of, but essentially you have your tissue sample and a capture slide. So your tissue sample is H&E stained. So you have this reference image. You select a region of interest as the capture area is only 6.5 by 6.5 millimeters. So you essentially need to select the area of interest from your tissue section. And then, but the beauty is that you look at the entire human transcriptome for Visium HD. What you then do is you permeabilize your tissue sample, and this is done on the CytAssist instrument, and then that allows for release of your of RNA onto the capture slide. So what you end up with is your H&E sample slide, and then you capture the RNA transcript information from that sample slide. This is our workflow. Sorry, and I just want to move this out the way. Okay, here we go. So essentially on the first day we do our tissue QC, which is we measure the RNA quality in your wax block sample, and this is done by DV200 score. If the sample passes, we then section samples. So the QC is done on a 10 micron section. If this passes, we then section 5 micron sections, which are captured on a normal negatively charged slide. These are then baked for 3 hours at 42 degrees and then stored in a desiccated room temperature for up to 6 months. So this is all part of the Visium HD protocol, but it's a nice way of going through, validating your samples and getting them ready for a run. So you can kind of stockpile a few samples and get them ready for the workflow. So in the next 2 to 3 days, depending on how vigorously you need the data or how quickly you need the data, you can then process your samples up to the pre-amplification step, and this is a SPRI select protocol, and you can store the samples at this stage for up to 4 weeks at -20, but generally we continue to doing the library preparation, and here you can store the samples at long term at -20. So this generally takes us into days up to day 4, essentially, in our workflow. And then in theory, you can do your sequencing still in the same week. So for example, if we were to run only 2 samples, so a single Visium HD capture slide captures 2 samples. If you were to process that all the way to sequencing, you could do the entire workflow in 5 working days. So that is a comfortable workflow, and it is possible. So in terms of the number of samples that you have to process, you can select different, this is the Illumina sequencing flow cell kits. You can select different flow cells, so either the P2, P3, or P4. And I've just included here roughly how many samples you'd be able to run. So on the P2, you'd be able to run 2 samples. P3 would be up to 7 samples, and P4 would be up to 10 samples. So we've tried each of these flow cells and with the numbers stated here, and we've gotten data from each run. So in terms of the sequencing depth, this does seem to be sufficient. So I just wanted to go through the QC metrics that we look at in terms of for us to select a sample. So this is the 10x recommendation, is to do a DV200 score on your samples. So this means that you take a 10 micron section from your wax block. They do recommend, if it is a wax block that has been sectioned before, that you do section a little bit deeper into the sample to make sure you are sort of reaching unexposed samples. If it's a completely fresh wax block, then you polish your sample until you get into the tissue, and then you take a 10 micron section. This 10 micron section is then processed according to the Quiogen kits that I've got displayed here. And basically, you isolating the RNA from that wax block section. And what you're wanting to look at is the RNA fragments that are greater than 200 nucleotides in size. And you look at a cutoff of at least 30% of them being greater than 200 base pairs in size. Because the Visium HD is a probe-based technology, it allows for detection of degraded RNA fragments. So you're no longer looking at, or in this case, you're not looking at the RIN score, but you're looking at the DV200 score. So measuring fragmented RNA. And then just in terms of our workflow again, depending on the sample size, so in terms of our lung tissues, they're usually quite big samples. So they're usually bigger than 1 centimeter squared. So I usually only take 1 or 2 10 micron sections per wax block. I found that 1 10 micron section is sufficient. But in terms of the gut biopsy samples, we've taken up to 4 sections because they only pinches essentially in each wax block. We've taken up to 4 10 micron sections just to test the RNA quality. So these are just some examples of the DV200 scores. So you run the extracted RNA on a tape station, and then you're looking at the RNA that's greater than 200 base pairs in size. So this percentage of total RNA. So this is an example of a sample that did not pass the QC metric, and this is an example of a sample that did pass. So the sample, for example, had a score of 36%. And then we have samples that have been collected from as far back as 2014. So I ran a whole bunch of samples, and this is just an idea of sort of the DV200 metrics that we have been getting. So this is not sorted by, age of sample. It's just a matter of, I think looking at each sample independently and making sure that it passes the DV200. So I don't think it's a matter of how fresh the sample is. It could be a matter of how the sample was prepared, how long it was in fixative for. Anyway, all of these contribute to RNA quality, and so it's really important to look at the DV200 metric. So at the top here, I just wanted to show this is the 10x recommendation of the DV200 metric, so looking at the DV200 score as a percentage, and then the mean UMI readouts per 8 micron bin. So it shows here quite nicely of, as you increase in the DV200 score, you're getting better and better transcript readouts. And then this is our sort of real, I was going to say real life data, but our lab data. So this is looking at lung samples. So you will notice that we get a lot lower UMI score per 8 micron bin. So we did have discussions with 10x, and it is a sample dependent metric. And for example, the gut samples, so this is the TMA example that I've shown, for those TMAs, we get a much higher mean UMI per 8 micron bin. So this is for a DV200 of 60%, you get roughly 100 UMIs per bin. And for our 60% lung sample, we get about 30 UMI per 8 micron bin. So the data is still really good and it is usable. And I'll show some of the example data now. One of the things we do want to try in the lab is one of the recommendations from 10x is to consider a different extraction temperature. So this is something that decross-linking temperature. So this is something that we will try in our workflow, and just to see if we get a higher RNA yield from the samples. So in terms of our second step in the workflow, we do sectioning, and we do a control H&E where we just look at how well the sample actually adheres to the slides. So this would obviously be a criteria where we exclude the samples if they don't adhere. And with TB lungs, very often the granuloma are very necrotic and they don't stick very well to the glass slides. So it's a stage at which we do exclude samples even if they do pass our DV200 metric. So that's unfortunately just the reality. So now I have an example here of a lung patient sample that we looked at. So this is the H&E section, and you do look at the area covered on the capture slide. So you can do this usually on the slide viewing software. And for example, here we see that it's an 87% coverage of the area. So the big square being the 6.5 by 6.5 millimeter area. And then this was the DV200 metric that we had. So this sample scored about 63%. So it's the best sample we had so far. This is the CytAssist capture with the fiducial frames on the outside. And hereafter you do the amplification of the RNA, and you do a SPRI selection cleanup. And just what's really important here to show is that the size of the amplicon is around 250 base pairs to 260 base pairs. And what's really important is that you want a very low CQ value when you do the qPCR on your isolated, on your amplified library. You want a very low CQ value. And also very important is that you have a very clean SPRI selected peak. So you don't want any smaller size fragments in this purified sample. And then we sequence these on our Lumina platform, and the Space Ranger pipeline now allows for single cell resolution. So this is really nice and really user friendly. So as far as I understand, it is still a Stardust based nuclear isolation protocol, and then it has a nuclear expansion associated with that. So that's how the single cells are selected from your H&E stain slide. So this is then each individual transcript mapped to that single cell bin, essentially. And this is the UMAP associated with the sample. So you get really nice separation of the populations and nice clean data. And then I did want to show, this is for the gut biopsy sample. The beauty here is that you can include four different, well, in this case, four different PIDs. Because the gut biopsy pinches are so small, you obviously don't want to waste the Visium capture slide on just a single gut biopsy, so we include them into these tissue microarrays. And you can buy molds to prepare your wax blocks for this. And then you just use biopsy punches to punch out your samples from the initial wax block to then prepare this tissue microarray. Yeah, and this is the data that we got from this particular sample, so also a very good DV200 score. So this is the CytAssist capture, and then again, the library preparation was nice and clean, no smaller fragments in the sample. And then again, this is the single cell resolution of the data. So it's really nice in this case. Each of these patients had undergone different treatment times of ART, and therefore we can essentially compare different treatment times and look at different cell populations or phenotypes expressed in these different gut biopsies. And then just going back to the TB sample slide, so this is the single cell resolution. It obviously allows for looking at just individual cell markers. So I'm interested in B cells. So just homing in on this particular granuloma within the lung tissue sample, we can look at expression of different cell markers. So CD20 is a B cell marker, so we see these interesting clusters associated with granuloma. CD68 is a macrophage marker, so macrophages are really important in sort of containing TB. And so we find macrophages present within this granuloma. And then CD4 T cells are also present around the granuloma tissue. And then what I'm interested in looking at is antigen presentation. So there are antigen presenting cells within these clusters. And then I was also looking at the interaction between B cells and T cells. So CCR7 is an important marker to look at here. And just to indicate that, yeah, the cells in these clusters are expressing CCR7, suggesting that B cells and T cells are coming together within these follicles. So finally, another way of looking at it is you can look at co-expression of different markers or different transcripts. So what I looked at here is essentially just different B cell activation markers. So you can combine a whole list of transcripts that are associated with B cell activation. So that's what I've done here. So the B cells are then stained in blue. And then I've got canonical B cell phenotypic markers that are expressed, and those are expressed on the cells in yellow. And then if they are co-expressed, the cells then stain green. So you can see again that within this is the granuloma again that we were looking at just now. Within those follicles, you see green stained B cells, suggesting that the B cells are activated within those follicles. One of the aspects we're looking at is whether or not B cells can present antigen to T cells. So here again, we see that the B cells within the follicles are staining green, suggesting that they are co-expressing B cell phenotype as well as antigen presentation. And then finally, we were looking at T cell activation within these follicles as well. And there are a few activated T cells in here. So it's just to highlight how you can, well, how we are looking at the data. Still quite preliminary, but it is data that we are generating, which we're very excited about. And yeah, and then that's just to thank all of our funders and everyone in the lab, all of the patients from our lung cohort, and then obviously everyone in the audience.